starting with the next
Next
one.
one,
Application of iron oxide nanoparticle in blood
blood
cell
cell
extraction.
extraction.
So
So
in
in
this
this
lecture,
lecture,
our
our
focus
focus
will
will
entirely
entirely
be
be
on
on
the
the
blood
blood
cell
cell
extraction.
extraction.
Uh,
Uh, uh, the meaning for this is, suppose if you want to ask me that, what is the meaning for this? Why we need to extract the blood cell? Uh, 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 in the testing of the blood, sometimes you need the blood cell separated from the plasma, and the conventional way of separating the blood cell is the centrifugation process. If you should centrifuge the whole blood, and you can separate the blood cell from that of plasma. We are going to provide a very easier solution rather than going through the centrifugation process. So the uh, 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 iron oxide nanoparticle provides a very viable solution to picking the blood cell from the whole blood, leaving behind the plasma. While performing the extraction, you have to be very careful that you are not supposed to break the blood cell, because when you are going to use the nanoparticle is very tiny in comparison to the blood cell. If the nanoparticle is going to interact with the blood cell membrane, they can break the blood cell and induce the hemolysis. So therefore, it means that we need a specific kind of iron oxide nanoparticles coated with some kind of biomolecules which have least affinity to interact with the blood cell to disrupt the blood cell membrane, but they can attach to the blood cell membrane without disrupting it, and in this way we can extract the blood cell. So this is the whole story on which this lecture is focused. We are using this same surface active magnetic nanoparticle which I here, the size close to 20 nanometer and coated with some kind of molecule.
what we do? So
So
we
we
have
have
two
two
kinds,
kinds,
obviously,
obviously,
the
the
surface
surface
active
active
and
and
the
water
water
soluble
soluble
magnetic
magnetic nanoparticle.
nanoparticle.
If you
If you
only
only
look
look
at
at
this
this
sketch,
sketch,
the
the
first
first
one,
one,
surface
surface
active
active
magnetic
magnetic
nanoparticles
nanoparticles
are
are
arranged
arranged
at
at
the
the
surface,
surface,
air
air
aqueous
aqueous
interface
interface
surface,
surface,
whereas the
whereas the
water
water
solubles
solubles
are
are
dispersed
dispersed
throughout
throughout
the
the
bulk
bulk
of
of
the
the
water.
water.
So
So
what
what
we
we
can
can
do,
do,
if I'm
I'm
going
going
to
to
place
place
the
the
surface
surface
active
active
magnetic
magnetic
nanoparticle
nanoparticle
on the
on the
whole
whole
blood cell suspension, and likewise I place the blood cell suspension in the
the
water
water
soluble
soluble
magnetic
magnetic
nanoparticle
nanoparticle
solution.
solution.
And
And
then
then
what I
what I
see is
see
that
that
if
if
I'm
I'm
going
going
to
to
and
and
let it
for
for
certain
certain
period
period of
of
time,
time,
maybe
maybe
few
2 hours,
hours,
you
you
can
can say.
say,
After
after
that,
that
I'm
I'm
going
going
to
to
attach
attach
the magnet, and I could see that when I attach the magnet, it's not only the magnetic nanoparticles are attached to the magnet, but magnetic nanoparticles are bringing the blood cells as well. So you
so you
could
could
see
see
that I
that
have
I have
taken
taken
out the
out the
blood
blood
cells.
cells.
And
And
same
same
can
can
happen
happen
over
over
here.
here.
You
You
see
see
the
the
arrow.
arrow.
This
This
arrow is
arrow is
going
going
over
over
here.
here.
This
This
arrow is
arrow is
also
also
going to
going to
this one.
this one.
But
But
then
then
apart
apart
from
from
that,
that,
we could
we could
see
see
that there
that
is
there is
another
an
arrow
arrow
which is
which is
coming
coming
over
over
here
here
and
and
another
another
arrow
arrow
from here.
here.
Apart from this sample, we
Sample,
have
we have
another
another
sample
sample
which
which
shows
shows
that this is
this is
one way
one
of
way of
extracting it.
extracting it.
While
While
performing
performing
this
this
extraction,
extraction,
you
you
may
may
break
break
the
the
blood
blood
cell
cell
and
and
create
create
a
a
red
red
color
color
solution
solution
because
because
of the
of the
oozing of
oozing
the
of the
hemoglobin
hemoglobin
from the
from the
blood
blood
cell,
cell,
and
and
that
that
creates
creates
the
the
red
red
color
color
solution.
solution.
Now,
Now,
the
the
amount
amount
of
of
the
the
red
red
color
color
solution is
solution is
proportional
proportional
to
to
the
the
amount of the
of the
blood
blood
cell
cell
broken.
broken.
Amount
Amount
of
of
the
the
red
red
color
color
solution
solution
that
that
is
is
created,
created,
that
that
is
is
proportional
proportional
to
to
the
the
number of
number of
blood
blood
cells
cells
broken.
broken.
So
So
this is
this
the
is the
whole
whole
story,
story,
and it
and
is
it is
being
being
tested with
tested with.
hemolysis. So I
So
mean
I
to
mean to
say
say,
that we
we
perform the
perform the
UV
UV
analysis
analysis
to
to
uh, figure
figure
out
out
how
how
many
many
blood
blood
cells
cells
are
are
being
being
broken
broken
while
while
performing
performing
this
this
extraction.
extraction.
But the
But the
best
best
magnetic
magnetic
nanoparticles are
nanoparticles are
those
those
which
which
can
can
extract the blood cells without
without
breaking.
taking.
So we
So
have
we
to
have
find
to
out
find out
the
the
which
which
are
are
the
the
best
best
magnetic
magnetic
nanoparticles
nanoparticles
can be
can be
used in
used in
order to
order to
extract the
extract the
blood
blood.
cell. So
So
this
this This schematic representation is only showing you the experiments we performed with the surface active as well as the water soluble magnetic nanoparticle. Here is the extraction photos for the extraction of surface active magnetic nanoparticle. This surface active magnetic nanoparticle soluble in hexane. We place the hexane suspended water surface active magnetic nanoparticle on the whole blood suspension. So this is blood suspension. We place them over here. Okay. Now, hexane is not solid in the aqueous phase. Hexane remains in the organic phase, and you have plenty of blood cells over here. The nanoparticles at the interface they will start interacting with the blood cells. Now, uh, after incubating it at 37 degrees centigrade for a few hours, place the you see the magnet over here. We place the magnet and trap and extract all the magnetic nanoparticles. So those magnetic nanoparticles we extracted, we see that there is one possibility. If the blood cells are there, then when we are going to put it in pure water, this sample we are going to put it in pure water, blood cells are going to burst in pure water because pure water is not a saline with not actually it is a buffer solution. So therefore, because of the osmosis, the blood cells are going to be burst, and you will have a colored solution because of the hemoglobin. The second one over here, the same concentration of the hemoglobin over here as that is over here. So what you can do, so you can make out that there is no extraction. So we can figure out by performing all these steps that if extraction has taken place or not. In this particular case, what we have observed that the hemoglobin is the hemoglobin absorbance is over here. This is the control, but this is the one from the sample. So practically there is no hemoglobin and hence there is no extraction of the blood cell. So all the blood cells are there in the solution, not picked up by the magnetic particle. But the magnetic nanoparticle has induced some sort of hemolysis. They actually break the cell membrane, and hence, uh, in this case, when we are going to have the sample from this part of the solution, so we have a small absorbance. This one is a control. This one is a small absorbance. So it means that interface at the interface when the magnetic nanoparticles are placed, so we may not have entirely facilitating extraction of the blood cell, and rather it is the magnetic nanoparticle involved in breaking the blood cell. So that is not our purpose. We don't want to break the blood cell. We have the other option then. What we did here, we use the water soluble magnetic nanoparticle. So here you see the water soluble magnetic nanoparticle. And in the water soluble magnetic nanoparticle, we picked and placed in the blood suspension. This is magnetic nanoparticle is the blood suspension. Now you might be wondering why it's not the red color. Because we have added magnetic nanoparticles, so that is why it's not red color. It's a dark color. Now, and eventually when we are going to place the magnet here, we have seen that when we place the magnet and we take out the magnetic nanoparticle and we put it in the solution to see if there is any hemolysis. If there is no hemolysis, it means that no blood cells are broken. Right. So here we have taken that there is some degree of hemolysis. That's why it's a colored solution. Then uh, the same experiment performed in another set of iron oxide. Now, what is the meaning of these two sets of experiments? In fact, they look identical. Whatsoever you see over here, the steps you see over here. But what is different? Different is this experiments were performed by magnetic nanoparticles stabilized with zwitterionic surfactant DPS, TPS, HPS. Alanine is amino acid, glycine is amino acid, histidine is lysine and valine. So what is the meaning of this? The meaning of is that we produce the magnetic nanoparticle by using a amphiphilic molecule as a colloidal stabilizer. As I told you that the magnetic nanoparticle can be stabilized with, uh, suppose if I say that this is a magnetic nanoparticle, okay? So this magnetic nanoparticle is stabilized by a bilayer of amphiphilic molecules. So this is the monolayer, and then we have the bilayer. Now, this amphiphilic molecule could be Gemini surfactant, conventional surfactant, amino acid. It could be amino acid as well. So these amino acids. But the amino acids usually they do not look like this because you do not find a much bigger non-polar functional group. But if you pick the non-polar amino acids, predominantly non-polar, you may have the same kind of amphiphilic properties as you could find with some other molecules. So in this way, amino acid molecule can also provide the colloidal stability, right? And in this way, we can use this as a, but it is not subjective. This is water soluble. Water soluble. When we place such kind of nanoparticle in the blood suspension, and place the magnet over here, magnet, right? We want to see that how many blood cells are going to be extracted by the magnetic nanoparticle by using the magnet. And the blood cells remain intact, they do not break. So here are the results. So we detect the breaking of the blood cell from the hemolysis. How much how much hemoglobin protein has oozed out in the solution? That is hemolysis. Hemolysis is the process by which the blood cells get break, and the hemoglobin which they contain, they that hemoglobin is oozed out. So that process is known as hemolysis. In simple word, it's the breaking of the blood cell and producing the hemoglobin. So here you have the hemolysis and hemolysis, right? What we observe that when we use all these amphiphilic molecules to create different kinds of magnetic nanoparticle. How many? One, two, three, four, five, six, seven, 8. We took different kinds of magnetic nanoparticle paired with different kind of amphiphilic molecules. It means that we have 8 samples of the magnetic nanoparticle. When we placed all 8 samples separately in the whole blood suspension, all 8 suspensions were able to break the blood cell. The breaking was up to 80%, close to more than 70% for the nanoparticles which were stabilized with glycine and alanine, and you could say close to 60% for histidine and valine, lysine and valine. Which is the least one? Least one are the zwitteronic surfactant. This is the zwitteronic surfactant. I have shown only the abbreviated form of the name of the zwitteronic surfactant. Zwitteronic surfactant, why I took the zwitteronic surfactant? The reason is that the zwitteronic surfactant with the polar head group contains both the amino as well as the acid group because there is zwitteronic and there is hydrocarbon tail. This is the zwitteronic surfactant. Now you see that this zwitteronic configuration chemically identically it's identical to that of amino acids. So that was the reason to compare because the amino acids are also zwitteronic, right? So therefore we took the amino acids
actual molecule, and the amino acid derivatized surfactant, we call this zwitterionic surfactant. We observe that if we choose to prepare the magnetic nanoparticle with only
only
amino
amino
acids,
acids,
they are
they are
highly
highly
toxic
toxic
to
to
the
the
blood
blood
cell.
cell.
This is
This is
very
very
strange.
strange.
Amino
Amino
acid
acid
functionalized
functionalized iron oxide nanoparticle. If I have a shell of
only
only
amino
amino
acid
acid
molecules,
molecules,
and
and
now
now
somebody
somebody
asks
asks
you,
you,
this
this
is
is
very
very
strange.
strange,
Amino
amino acid
acid molecule shell is going to
to
break
break
the
the
blood
blood
cell.
cell.
So
So
suppose
suppose
if you
if you
want
want
to
to
see
see
that
that
how
how
it
it
is
is
happening at
happening at
microscopic
microscopic
level,
level,
I
I
say
say
this is
this
the
is the
blood cell.
cell.
Okay,
Okay,
it's a
it's a
disc
disc
shape.
shape.
This is
This
the
is the
blood
blood
membrane.
membrane.
It's
It's
a
a
lipid
lipid
membrane most of the time, right? Means it is predominantly containing lipid molecules. It's a cell wall, right? And if I have the magnetic nanoparticle coated with amino acid, and it means that amino acid has the ability to puncture or to break the cell wall. That is why the hemoglobin that is over here, so that will be oozed out to create the hemolysis. Am I correct? Now, so, okay, so now we will try to understand that what is the chemical composition. Chemical composition of the blood cell membrane. I'll show it in the few slides after this one. But just
just
schematically, I can show you
show you
what
what
is
is
happening.
happening.
The cell
The
wall
wall
is in fact, it's a kind of a structure where you have the head groups because you have the lipid molecules, so they have the two hydrocarbon tails. So lipid molecules are creating the cell wall. This is the cell wall. Okay, cell wall. It's not only the lipid molecules, there are other molecules as well. You will see there are membrane proteins as
as
well.
well.
Now,
Now,
this
this
is
is
the
the
head
head
group
group
of the
of the
lipid
lipid
molecule.
molecule.
Most of
Most
the
of the
time it
time
is
it is
zwitterionic
zwitterionic
or
or
maybe
maybe
anionic,
anionic.
right?
Right?
If I
If I
say
say
that
that
okay,
okay,
it is
it is
zwitterionic,
zwitterionic,
and
and
now
now
I
I
have
have
the
the
magnetic
magnetic
nanoparticle,
nanoparticle,
and
and
it
it
is
is
coated
coated
with
with
a
a
bilayer
bilayer
of
of
amino
amino
acid.
acid.
So
So
amino
amino
acid is
acid is
also
also
like
like
this,
this,
plus
plus
and
and
minus.
minus.
It
It
means
means
that
that
I
I
am
am
initiating the
initiating the
electrostatic
electrostatic
interactions
interactions
between
between
the
the
positive
positive
and the
and the
head
head
group
group
with
with the with
the
the
acid
acid
group
group
and
and
the
the
negative
negative
end of
end
the
of
head
the head
group
group
with
with
amino
amino group,
ammonium
ammonium
group.
group.
Right?
Right?
So in
So in
this way,
this way,
the
the
electrostatic
electrostatic
interactions
interactions
will
will
disturb
disturb
the
the
cell
cell
membrane
membrane
and
and
hence
hence
it
it
will
will
break
break
it
it
and
and
leading
leading
to the
to the
oozing
oozing
out
out
of
of
the
the
hemoglobin.
hemoglobin.
So
So
this is
this is
happening
happening
in
in
the
the
case of
case of
amino
amino
acids.
acids.
But
But
why it is
it's
not
not
happening to
happening to
that extent
extent
when we
when we
use,
use,
you
you
see the
see the
blue color? Blue
blue
color is
color is
that of
that of
DPS,
DPS,
it's a
it's a
C12.
C12.
So
So
when
when
the
the
docile,
docile,
when this
when this
hydrocarbon tail is
dot,
docile, then
then
it is
it
DPS. When it is C14, it is tetradecyl, it is TPS. When it is C16, it is hexadecyl HPS. So the
the fact is, the fact is, obviously less than half deuteranic surfactant in comparison to the amino acid. It means that somehow the zwitterionic surfactant they have less, though they are also breaking
the
the
cell
cell
wall,
wall,
but
but
not
not
up
up
to
to
that
that
extent. On the other hand, if I take cyclodextrin, Do you familiar with the cyclodextrin? Cyclodextrin is very,
It's a polysaccharide. It's a cyclic saccharide molecule. It's a ring-shaped structure, right? And if I take the derivatives of sugar, cyclodextrin, alpha, beta, dextrose, lactose, or sucrose, then there is no hemolysis except for this one, sucrose.
they are not doing any
Any
breaking
breaking
of
of
the
the
cell
cell
wall,
wall,
right?
right?
And
And
it
it
means
means
that
that the
uh, uh,
uh,
uh,
uh,
the
the
most of
most
the
of the
blood
blood
cells
cells
are
are
extracted.
extracted.
So
So
here
here
there is
there
the
is a
least
least
extraction,
extraction,
maximum
maximum breaking.
here
Here
least
least
breaking
breaking
and
and
maximum extraction, right? So here how many samples we use? 1, 2, 3, 4, 5. So we have 5 samples of sugar
sugar
that
derivatized molecules which were providing the colloidal stability to the iron oxide nanoparticles. And what we observed that they are working excellently and they are extracting the blood cell without inducing the hemolysis. These are the experimental observations. So I'm taking you to the next slide. Now, where is the breaking of the blood cell? So these are the blood cells. You see the pictures? Where are the broken blood cells? See the puncturing, the holes in the blood cell, right? So the holes in the blood cell showing that
when the
when the
nanoparticle
nanoparticle
is
is
interacting
interacting
with the
with the
cell
cell
wall
wall
and
and
it
it
is
is
breaking the
breaking the
cell
cell
wall,
wall,
the
the
hemoglobin
hemoglobin
is
is
taken
taken
out,
out,
right?
right?
And
And
now
now
blood
blood
cell is
cell is
broken.
broken.
On the
On
other
the other
hand,
hand,
you
you
see
see
this
this
blood
blood
cell,
cell,
and
and
you
you
see
see
these
these
are
are
the magnetic
magnetic
nanoparticles.
nanoparticles.
You
You
see
see
all
all
these
these
are the
are the
magnetic
magnetic
nanoparticles.
nanoparticles.
Magnetic
Magnetic
nanoparticles are
nanoparticles are
attached
attached
to
to
the
the
cell
cell
wall.
wall.
You
You
won't
won't
expect just
just
1,
one,
2, or
two, or
few
few
nanoparticles.
nanoparticle.
There
There
is
is
plenty of
plenty of
nanoparticles
nanoparticle.
those are
Those are
going to
going to
attach
attach to the
to the blood
blood
cell
cell
membrane.
membrane.
So
So
here
here
you
you
have
have
so
so
many
many
blood
blood
cells,
cells,
all
all covered with the magnetic nanoparticles. So here you also have, so you could see here, it's not clear because it's a 5 micron. You see the range? It's a very big range. But here it is 500 nanometers, so that is why you could see nanoparticle. When the nanoparticle size is too tiny, only it is 20 or 30 nanometer, whereas the blood cell is micron, 5 to 6 micron in size. So there's a huge disparity between the size of the blood cell and magnetic nanoparticle. So that is why you may not see clear, but only you could see that all these are the magnetic nanoparticles. We can perform the matching and you see proof. So this area
is
actually
covered
covered
by
by
the iron oxide nanoparticle. This is the mapping for the iron, this is the mapping for the oxygen to confirm that indeed all the particles which are present, they are made up of iron oxide. Okay, so this is the way we see. This slide is extraction and blood cell morphology. So humanoid cells. So here we have shown you these particular two images. So they are showing you the humanized broken blood cells. But you would like to see the unbroken
broken
blood
blood
cells.
cells.
See
See
over
over
here.
here. You see clearly the blood cell?
The disc-shaped blood
Cell?
cells. These cells
Cells
are
are
not
not
in
in
disc
disc
shape
shape
because
because their
their morphology
morphology
somehow
somehow,
altered. It
it
means
means
that
that
all blood cells
is
in this sample, they were interacting with the magnetic nanoparticle, but in most of the cases, the cell membrane got punctured or disturbed, broken, and some cases, the morphology of the blood cell from the disc shape has been changed. It's not a disc shape. That what is a normal blood cell shape is this one. You see the bowl shape, disc shape, it is the disc shape blood cell. Now, you see the disc shape, all these blood cells are intact. They are in its natural shape
shape
and
and
structure.
structure.
No
No
alteration
alteration
in the in the
in the
structure
structure
of
of
these
these
blood cells.
cells. So extraction and blood cell morphology. So here non-humanized cell. So this extraction is the best extraction where the morphology of the blood cell has not been altered. So therefore, if you ask me that which were the successful experiment, these were the successful experiment. So it means that the images where you see the intact blood cell, those were images of these samples. So these samples where no hemolysis has taken place. You see over here, this color is why this color. Most of the blood cells are extracted over here, right? When you put this sample in pure water, the blood cells got burst and created the blood. Right.
On the other
On the other
hand,
hand,
if we
take
take this
this.
one and put it in pure water, there is no color. What's the meaning for this? There are no left blood cells in this sample. That's why there is no color. Here, all blood cells are extracted intact. So when I put them in pure water, they are going to burst. So the intensity of the color is corresponding to the number of blood cells extracted. As more and more color is there, more and more blood cells are extracted. And that's what you are seeing in the image.
Then, what is the conclusion for these experiments? How can we use this technique to replace the centrifugation method? What we should do? We should come to the mechanism. Here is the mechanism. So I have divided this into a complicated image. I'm going to explain it. Okay, first I'm going to show this is the morphology, molecular arrangement of the blood cell membrane. This is lipid bilayer. This is the surface of the cell. Outside is water. No.
Right, outside is the aqueous phase. Here, inside is the blood cell. So I'm showing this the surface of the blood cell. And the membrane of the blood cell is
Mainly composed of
that is a lipid bilayer. See the structure of the molecule? These
these
are
are
the
the.
lipid bilayers. Okay, in this lipid bilayers, uh,
Actually, I have not labeled many other things here. So this one is the membrane protein. You know the membrane protein that provides the reinforcement to the cell wall structure. That is something like this. Okay, so then there are other components also. Okay, I should have labeled them, but anyway, apart from this, you have the glycolipid molecules.
these
These
are the
go
glycolipid molecules. Okay, and these are the
uh,
sugar derivatives, sugar molecules, and below the cell membranes, immediately below the cell membranes, you have the spectrum. It is the arrangement of the membrane
membrane
skeletal
skeletal
structure.
structure. It provides the support to the membrane of the blood cell. Okay, what happens now? Well, I have divided. Initially, you concentrate on the cell wall and the but actually the cell wall is in action rather than any kind of involvement of spectrum.
So it is the lipid bilayer membrane, so that is in action or interacting with the magnetic particles. I have divided the diagram into two parts. This part belongs to the hemolyzed where
Where
the
the
hemolysis
hemolysis
has
has
taken
taken
place.
place.
Hemolysis
Hemolysis
has
has
taken
taken
place, it means the
place.
membrane has broken down and the protein has been installed. Right, this part belongs to where no hemolysis has taken place. See over here. Hemolyzed blood cell, non-hemolyzed blood cell. Now, microscopically, what happens? See over here, when I am going to take the amino acid stabilized magnetic
Magnetic
nanoparticle,
nanoparticle.
so they are
They are
stabilized like this. I'm
using the
using the same
same
kind of
kind
a
of a
picture
picture
over
over
here.
here.
The
The
bilayer
layer
or
or
the
the
coating
coating
of
of
the
the
amino
amino
acid.
acid.
Right? When they
When they
come in
come in
close,
close,
in
in
contact
contact
with the
with the
cell
cell
membrane,
membrane,
these
these
are
are
the
the
head
head
groups
groups
of
of
the
the
phospholipid molecules.
phospholipids.
It's the
It's the
same
same
kind
kind
of interaction which I showed
of
you.
over here.
The head group of the phospholipid molecule and this is the dipolar amino acid. So the electrostatic interactions are taking place, with the result of which this membrane is disrupted and hemolysis is set in. The protein is oozed out. And therefore, these nanoparticles, which are coated with the protein, or these are the biological surfactants, even biological surfactants are not working. They also have the ability, to break the cell membrane, and we can use for the extraction of
A
blood
blood
cells.
cell.
So
So
it
it
means
means
that
that
these
these
magnetic
magnetic
nanoparticles,
nanoparticles, which are produced with
various
this
kinds
kind
of
of
amino
amino
acids
acids
and
and
the
the
biological
biological
surfactant,
surfactant,
they are
they are
not
not
good
good
magnetic
magnetic
nanoparticles
nanoparticles
for
for
extracting the
extracting the
blood
blood
cells.
cells.
But
But
what
what
are the
is
good?
good?
Cyclodextrin. Cyclodextrin is, let me just show you the cyclodextrin
Cyclodextrin
is
is
kind of
kind
a
of a
structure
structure
like
like
this.
this.
It's created by the the
Aromatic
aromatic rings of the sugar,
sugar,
aromatic rings of the
the
sugar,
sugar,
and there is
this
an empty
empty
space over here. It is non-polar from inside, polar from the surface because of the hydroxyl groups. Right, that is the cyclodextrin. I have shown this cyclodextrin in the its
bucket
bucket-shaped
shape,
molecule. You see the bucket-shaped
shape
molecule.
molecule.
The
Bucket
bucket-shaped
shape
molecules
molecules
have
they
arranged on the
on the
surface
surface
of
of
magnetic nanoparticle.
nanoparticle,
Right,
right?
and they
And
are
they are
providing the
providing the
colloidal
colloidal
stability.
stability.
They are amphiphilic molecules, so therefore
Therefore,
they
they
can
can
be
be
used
used
as a
as a
colloidal
colloidal
stabilizer.
stabilizer.
What is
What is
happening?
happening?
This
This bucket
bucket-shaped
shape
molecule has a
molecule
cavity
cavity
inside.
inside.
The
The
glycoprotein,
glycoprotein,
which has the sugar
sugar,
entity, that can be incorporated into the bucket. This
this
cavity inside the
the
bucket,
bucket,
it
it
accommodates
accommodates
the
the
glycoprotein
glycoprotein
sugar entity,
entity.
and
And
because
because
of
of
the
the
non-polar
polar
interactions.
interaction,
So therefore,
therefore,
magnetic
particle
nanoparticle is not coming
coming
into the
into the
vicinity
vicinity
of
of
the
the
bilayer
bilayer
membrane.
membrane.
It
It
is
is
entangled
entangled
over
over
here.
here.
Magnetic
Magnetic
nanoparticles
nanoparticles
are
are
entangled
entangled
over
over
here,
here
and
and
they are not
directly
directly interacting
interacting with
with the bilayer membrane. They have also the ability to interact with the bilayer, but most of the magnetic nanoparticles do not interact with the blood cell membrane, though I have shown few of them. We cannot entirely exclude the possibility of interacting with the lipid bilayer, but most of the magnetic nanoparticles, they interact with the I say
say
the
the
glycoproteins,
glycoproteins,
with the
with the
result
result
of
of
which
which
the
the
membrane,
membrane,
bilayer
bilayer
membrane,
membrane,
remains
remains
undisturbed,
undisturbed,
and
and
hence
hence
the
the
anchoring
anchoring
of the
of the
magnetic
magnetic
nanoparticle,
nanoparticle.
there are
There are
several
several there are
several
several
magnetic
magnetic
nanoparticles.
nanoparticles.
Those
Those
are
are
going
going
to
to
anchor.
anchor.
You
You
see,
see,
several
several magnetic
magnetic nanoparticles
nanoparticles
anchoring
anchoring
to
to
just
just
one
one
blood cell.
cell.
And
And
hence,
hence,
when
when
you
you
are
are
going
going
to
to
apply
apply
the
the
magnet,
magnet,
placing
placing
the
the
magnet,
magnet, the magnet
is
is
attracting
attracting
the
the
magnetic
magnetic
nanoparticle
nanoparticle
because
because
blood
blood
cell
cell
is
is
not
not
magnetized,
magnetized,
right?
right?
So
So
when
when
you are
you are
applying
applying
the
the
magnet,
magnet,
the
the
whole
whole
assembly
assembly
is
is
dragged
dragged
towards the
towards the
magnet.
magnet.
This is
This
the
is the
extraction, the
extraction, the
whole
whole
assembly.
assembly.
Now, the meaning of
Of
this
this
is,
is
now,
now
this assembly contains not
not
only
only
the magnetic
magnetic nanoparticle,
but
but
also the
blood
blood cells. So when we are going to place the magnet over here, so a magnetic nanoparticle along with the blood cell, they are being extracted, and that is why they are over here. So in this way, what is the mechanism? The mechanism is suggesting us when you take the magnetic nanoparticle stabilized with sugar derivatives means which one? Cyclodextrin,
cyclodextrin,
alpha
alpha
beta,
beta,
dextrose,
dextrose,
lactose,
lactose,
may
may
not
not
be,
be,
sucrose
sucrose
is
is
not
not
good.
good.
So
So
if
if
you're
you're
going
going
to
to
stabilize
stabilize
with
with
these
these
sugar
sugar
derivatives,
derivatives,
then
then
they are
they are
very
very
good
good
carriers of
carriers of
blood
blood
cell.
cell.
They are
They are
very
very
good abstractors of
protectors.
blood cell. They are
They are
not going to
to
break the
break the
blood
blood
cell,
cell,
and
and
then you
then you
can
can
use
use
them
them
for
for
extracting
extracting
the
the
blood
blood
cell
cell
leaving
leaving
behind
behind
the
the
serum,
serum,
and
and without performing the centrifugation, you can simply take out the blood cell, and this is a new technology, new methodology, and you do not need any kind of mechanical centrifugation process to separate the blood cell. It's a very quick, simply put the blood cell, magnetic nanoparticle stabilized with cyclodextrin or the sugar in the whole blood cell, place the magnet, take it out, and you have the blood cells. So this is the meaning of the whole story. Okay, here is the summarization. So summarized all different kinds of magnetic nanoparticles we use.
Extraction versus hemolysis.
We want least hemolysis, maximum extraction. That is the meaning of the usefulness of the methodology we are producing. We want that least hemolysis being induced, and we want the little number of blood cells are broken, but most of the blood cells are extracted. Okay, so we have prepared all the different samples of blood cell. You see, 0 to 100% is the hemolysis. So in this case, this is the structure of cyclodextrin. As I told you, the ring-shaped structure. This is the cavity equated with the bucket-shaped structure. It is basically a bucket-shaped molecule and empty from inside. It's mainly non-polar from outside, right? So when we use dextrin, we also call oligosaccharides, means sugar molecules arranged in a cyclic arrangement. That is the meaning of oligosaccharides. So when we use such kind of molecule for the synthesis of the magnetic nanoparticle, there is the least hemolysis, maximum extraction. More than 50% of the extraction, right? Take dextrose. For that, effect is almost the same. Dextrose is also useful as much as cyclodextrin. Biological surfactant, sodium cholate. This is sodium cholate. If you look at the internet, it's a cholesterol derivative, biological surfactant, right? If you take sodium cholate, they're giving us any better results. So there is still higher hemolysis in comparison to this, right? But the extraction is much less, 50% than this. To zwitterionic surfactant. This is zwitterionic surfactant, amino acid, or other.
If you take surface active, if you take the surface active, all those are from here to here, they are the water soluble magnetic nanoparticle. Surface active magnetic nanoparticles are not working. So in the case of surface active magnetic nanoparticle, hemolysis is too much, extraction is very less. So this is the
Conclusion. And if you ask me that among all magnetic nanoparticles, which are the best performer, I would say that they are the sugar derivatized
stabilized
magnetic
magnetic nanoparticles are
the best one to extract the blood cell. So you may ask me that when next time you go for the blood test, would you expect that the people in the blood lab you using this kind of technology? I'm not sure at least immediately. Maybe after some time, probably they may use such kind of technology rather than centrifugation your blood sample to extract the blood cell and to perform the test and to test the serum and for any kind of other purposes. So, but this is a new kind of a technique, methodology can be easily used and is much easier to be used in comparison to the centrifugation. Conclusion, use surface active magnetic nanoparticle with less potential to disrupt blood cell membrane. Now, the surface active magnetic nanoparticle I have shown you, they are not good enough. They are not good enough for the extraction, but we need more experiments. Probably we may have some other kinds of surface active magnetic nanoparticle. They can induce less hemolysis and may prove better extractors, and they can work exactly the way we have shown you water soluble molecule. But for the time being not working. So they have the less potential to disturb blood cell membrane biological applications at interfaces. Now, uh, it has several implications. I mean to say that when you are using the iron oxide nanoparticle for hyperthermia, magnetic hypo, MRI, tissue engineering, you need the magnetic nanoparticle which do not disrupt the blood cell. So therefore, when you have to perform those kinds of experiments, you would prefer to use the magnetic nanoparticle which are not breaking the blood cells. So that is the meaning for that. Cyclodextrin, dextro, stabilized water-soluble magnetic nanoparticle are the best components of the water-soluble magnetic nanoparticle for bulk biological system or the extraction of the blood cell from the whole blood. So this is all about regarding blood cell direct application on which this topic was iron oxide nanoparticle for biomedical application, right? But you could see that in order to arrive at this concluding topic, I had to walk through so many lectures, right? If I could have directly come to this lecture, you could imagine now you have gone through all the lectures, how difficult it was for you to understand what is happening. So that is the one when we design the course, we always design the course. We start with the basic and slowly we go ahead to understand the complicated concepts. So that is the best way of understanding the science, right? Science is not a story where somebody is telling you the story and it's directly going into your brain. It is the evidence which you have to prove. It is the quantification which you have to measure. The evidence has to be quantified. So somebody can say that it's a weak evidence, strong evidence. It's not that way the science is being explained. Evidence means explained in terms of the numbers. We say this is the evidence, these are the numbers. To prove that is a strong evidence, that is known as the quantification. So we are left with just one lecture in the afternoon. So we will see you again. So thank you so much for your kind attention. You can have any question now. Thank you. Yes, please.
Has iron containing proteins. So, sir, why we can't use that things for the magnetic field? Why we put that nanoparticles?
You are asking, you are saying that the blood proteins containing iron.
Yes, sir.
Can, can you repeat your question once again?
Sir, blood proteins
Yeah,
contain iron proteins like
correct.
protein. So, sir, why means till now we use centrifugation for extraction, right? But then why we need nanoparticles?
Yeah. So that's a very valid question. So you know that the blood protein is a hemoglobin, and the heme group you already know. It's the in the center of the heme group there is iron that is coordinated, right? So she's talking about that iron. Now that iron is not magnetized. It cannot interact with the external magnet or the magnetic field. So here the purpose of the magnetic nanoparticle is to attach the magnetic nanoparticle to the blood cell. We can use any other nanoparticle. Suppose if you ask me, why can't we use the gold nanoparticle, silver nanoparticle for this kind of extraction? I would say that the silver or the gold nanoparticle may not be having that much advantage as much the iron oxide. The reason is they are not going to be attracted to the magnet. So here we are talking about the ease of the extraction. The ease is when the iron oxide nanoparticles attach to the blood cell, when we place the magnet, the whole assembly is attracted toward the magnet. If I am using gold nanoparticle instead of iron oxide, I may place the magnet they are not going to be extracted. Again, I have to use the centrifugation method. So in order to avoid the centrifugation method, iron oxide nanoparticles are used. They are attached to the cell wall. They are not breaking the blood cell, but they are attached. So therefore, when I apply the magnetic field, magnetic nanoparticles are bound to be attracted towards the magnetic field, but they are attached to the blood cell. They are dragging the blood cell towards the magnet. In this way, we are extracting them. So that is why it's the ease with which. So in when you have to use the application, or you use the magnetic or any other nanoparticle for any kind of application, it is the applicability, it is the ease with which we are going to apply it, so that the methodology which we are creating, that is much better methodology economically, time purposes, and with the ease of achieving the objectives of that methodology in comparison to the same methodology already existing. So extracting filter means, if somebody asks you that in what way this technique is better, you'll be saying that we need to extract the blood cell from the serum to perform certain tests to see if you have anemia, right? And there are other things you have to see, if other critical illness is related to the shape of the blood cells, right? So for all those purposes, if you have to remove the blood cell from the whole blood, leaving behind the serum, you will be performing time consuming centrifugation technique that is energy intensive as well as time consuming. But this one is not energy intensive. Simply picking the magnet, attaching, blood cells are separated out, and you can take out the blood cell and see through the microscope if their shape or the size is the same. Otherwise, you have the anemia. That is it.
Active nanoparticle, it's only dependent upon the surfactant molecule.
Correct. Absolutely. It is the
Gemini surfactant. It is not only one kind of Gemini, it is a whole class of molecules and several series of Gemini surfactant. It is your experience performing the experiment will teach you that which kind of Gemini surfactant is most suited to create the surface active magnetic nanoparticle. Not all Gemini surfactant, and just few of the conventional surfactant can lead to the surface active magnetic nanoparticle. But most of the Gemini surfactant can help you create the surface active magnetic nanoparticle. Is it okay? Any other question, please? Yes, please. Excuse me, please. Once again.
There was discussion about the incorporation of nanoparticles in films.
in films. Okay. Yeah. So you are asking me?
How does the incorporation of nanoparticles enhance their structural stability?
Yeah.
Okay, just a minute. Actually, there was another diagram which I've not included in the slide, so I'm going to draw it now. This is magnetic nanoparticle.
This is magnetic nanoparticle. Okay. The protein is something like this. Okay. And you have another protein that is something like this. So the proteins are adsorbed on the surface of the magnetic nanoparticle through hydrophilic domains. Predominantly, hydrophilic domains have to adsorb on the transition metal nanoparticle. So you may expect that this is the hydrophilic domain that is on
on the surface, acting as anchoring site to attach the protein to the nanoparticle. The same is the story over here. But where are the hydrophobic domains? Hydrophobic domains are away from the surface, and because of the known polarity, the
hydrophobic domains of this protein molecule,
hydro
hydrophobic domains of this protein molecule, they have the non-polar interactions. These non-polar hydrophobic interactions through the hydrophobic domains of adjoining
joining
protein
protein
molecules,
molecules, they are very intense, right? So therefore, you see that this protein macromolecule is attached to this because of the nanoparticle. Helps to bind the protein molecules and to create a reinforcement in the form of a protein film. Okay. Any other question?
Thank you so much, sir. Now there will be a short tea break for 15 minutes, after which there will be tutorial conducted by Dr.